Dr. Jason Saunders here, HBOT USA, and another research review. Today we're going to talk about hyperbaric oxygen and stem cells. So what is a stem cell? Basically a stem cell, some people call them naive cells, they're progenitor cells. They are basically undifferentiated cells. You can call them baby cells that haven't grown up yet. And so if you think about it like this, at one point in our development, we were literally two cells that came together to become a single cell and then divided many millions of times. And so ultimately, every cell in our body has the DNA to have become any other cell in our body. But once they differentiate, then they stay in that form. So once a liver cell becomes a liver cell, it stays a liver cell, and when it replicates, it makes more liver cells, and intestinal cells are the same way, and neurons would be the same way. And so, but our body renews itself periodically, and it renews itself through the use of stem cells. So we still have a storage form of immature, undifferentiated stem cells that over time can come out and help heal tissue, heal wounds, heal trauma, and so you could basically rebuild certain parts of our body. Now, some of our tissue regenerates more easily, more quickly. Some of our tissues regenerate very slowly, if at all. And so depending on the cell type determines the speed and the ability of our body to regenerate. But we regenerate through taking undifferentiated cells, differentiating them into that cell type, and then having them grow up to do the function of that tissue. So the question here is, there's a lot of interest in stem cells now because we're using them in so many different places. Stem cells for degenerative diseases, whether it's neurologic degenerative disease like brain or nervous system breaking down over time, orthopedic degeneration like hips and knees. So as joints are breaking down, we're using stem cells. For wound care, for all kinds of different issues, what we're trying to do is we're trying to harvest immature cells and then collect them and then, either IV or inject them, a couple different ways of getting it in, but getting these cells into our body, into a location of trauma or injury or degenerative or, arthritic type processes, and allowing those stem cells to then differentiate into that tissue, and with the idea, hopefully, that tissue can now regenerate and heal. So here's the thing. Hyperbaric also helps create stem cells. So there's two ways we could look at hyperbaric. We could look at hyperbaric from the standpoint of oxygen is a fuel that our cells need for function, and hyperbaric is a way that we can stimulate further stem cell release. So we're going to talk about that version first. There have been multiple studies on the process of prolonged oxygen exposure in a hyperbaric environment, releasing stem cells. Now, it's not just mesenchymal or bone marrow stem cells, also central nervous system stem cells. In other words, rebuilding brain tissue as well as the musculoskeletal system. So hyperbaric oxygen most likely through HIF1 exposure, hypoxic induction factor, triggers, or through some type of nitric oxide release, stimulates these mobilization of stem cells throughout our body, stimulates them to be released and then mobilized or moved into certain tissues that are in need of increased stem cell and repair. And so we know that prolonged, again, not as much short term. There was a particular study, which I'll post in the bottom in the description, that showed after a single HBOT session, CD34 increased dramatically. And CD34 is a certain blood marker that when it's increased is associated with the fact that our body is also releasing stem cells. So there are some studies to show that short term hyperbaric does stimulate some type of stem cell release, but primarily we're looking at somewhere between at least 20 to 40 sessions in order to get, a true increase in stem cell circulation. Depending on the study, some have found somewhere between 30 and 50% increase in stem cells. Some have found somewhere between 4 and 8 times the amount of stem cell release as a result of longer term hyperbaric oxygen exposures. So right on its own, with the addition of hyperbaric oxygen, it will stimulate a lot of healing. And as we stimulate healing, one of those processes will be stem cell release. So you could basically use your own stem cells that you have and expose yourself to this hyperbaric environment over a period of time and upregulate your own body's ability to release these stem cells into the places of need to produce that type of a healing effect. Put that aside for a minute. Another thing that we're looking at is, well, people are getting stem cell therapies, whether it's a stem cell injection, a stem cell IV, exosomes, which are, to simplify it, almost like stem cell instruction packets. So we're already utilizing this idea of whether it's our own stem cells or from other sources, but getting this increased level of stem cell deposited in some type of area of need to stimulate some type of healing response. Now, in order for cells to differentiate, they need to be placed in the right location, and they need the right signaling factors, and oxygen plays a huge role in that signaling process. So there are also handfuls of studies that are showing that, hyperbaric by itself is great and does X, Y, and Z. Stem cell therapy by itself is great and it does X, Y, and Z. But when we combine the two, when we use hyperbaric oxygen in combination with stem cell therapy, we get a magnification. Typically, it's very significant magnification of the combination of the two together when we're talking about this healing response. And so there are a few different protocols out there. There are a few different ones that we use. Primarily, to some degree, you'd be looking at some amount of hyperbaric therapy before your stem cell therapy, and the reason for that is really because, again, so much of this is relying on the environment being conducive to healing and the proper signals being in place to produce that healing effect. So by pre-exposing the person to hyperbaric oxygen, you're already going to reduce inflammation by many mechanisms. They're described in some of these other videos. So you're going to reduce inflammation. You're also going to increase the healing response. So you're going to start stimulating all of these signaling chemicals in our body, upregulating our own stem cells, upregulating our own regeneration chemicals to start that response. So now you're primed and you're actually ready for your stem cell therapy. So your injection, your IV, whatever you're going to end up getting. So now the area has been primed, it's deflamed, it's got the proper signals going, you're starting to release your own stem cells into the area. Now you're going to get an injection or an IV of an additional amount of stem cells. And then you're going to after the stem cell therapy, do more hyperbaric oxygen as a way to continue to feed those stem cells the oxygen, the nourishment that they need to continue that signaling effect, to continue to increase your own stem cell release, and also to continue to stimulate ATP production. ATP is the energy system of the cell. It's the energy currency of the cell. And so as you upregulate oxygen, you upregulate ATP production. You're giving those cells the energy that they need to differentiate into the proper tissue and to literally create the healing response that you're trying to create, both with the hyperbaric alone and with the stem cell therapy as well. So in summary, take a look at some of the studies below in the description, but basically 2 ways to look at this. Hyperbaric by itself, and in so many cases, especially if it's not a very advanced case of trauma or degeneration, hyperbaric oxygen therapy on its own over a prolonged period of time certainly has its own massive benefits, a lot of inflammation control, a lot of regeneration signaling, a lot of stem cell release that'll already start that process. In more advanced cases where you're really looking for an additional source of either exosomes or stem cells, then the idea here would be, well, create the right environment. So do some 10 to 20 hours of hyperbaric before your stem cell therapy. Start that process, start upregulating the healing response, start upregulating your own stem cell release. Prepare the body for that therapy. Once the body's prepared, get your stem cell therapy, get your injection, your IV, whatever you're going to do. let it simmer for a couple days, and then follow that up with the next round of hyperbaric therapy to continue signaling the healing response, continue releasing your own stem cells, to continue reducing inflammation, and then to continue to feed oxygen to those newly differentiated cells so that they're creating the amount of ATP they need, one, for their own differentiation,